severe weather, so that's why my voice is lost. But hopefully we can recover. Welcome today. We have a very special in-person guest. I follow Dr. Chen research for many, many years, and we're so delighted to have her here in person. So with that, let's Yashi to introduce our speaker.
Oh, okay, I touched it. Okay. Good morning, everyone. My name is Sarai Rios, and I'm a graduate student in Dr. Zoda's lab. It is my absolute pleasure to welcome you all to today's seminar. I am honored to introduce today's guest speaker, Dr. Chen, the Dr. Bernard and Janet R. Becker Distinguished Professor of Ophthalmology at the Washington University in St. Louis. Dr. Chen is a leading expert in vision science with their research focus on inherited retinal diseases, particularly CRX-linked retinopathies. Dr. Chen earned her BS in biology from Capital Normal University in Beijing, followed by an MS in biochemistry from Peking University Cancer Institute. She then pursued her PhD in biochemistry and molecular biology at SUNY Upstate Medical University and completed postdoctoral training in vision sciences at Johns Hopkins University. Her pioneering work has significantly advanced our understanding of photoreceptor gene regulation and retinal degeneration. Dr. Chen has classified CRX mutations into 4 distinct types and developed genetically engineered mouse models to study these mutations. Her team employs molecular genetics, functional genomics, and AAV-mediated gene therapy to explore treatment strategies for inherited retinal blindness. Most recently, she has expanded her research to include retinal organ cultures, creating miniature human retinas in a dish as models for disease study and therapy development. Beyond research, Dr. Chen plays a critical role in training the next generation of scientists. She serves as the co-director of the NIH T32 Interdisciplinary Training and Vision Science Program at Washington University and has mentored numerous trainees working in vision research. Her work has been continuously supported by the NIH and private foundations, including the Research to Prevent Blindness Stein Innovation Award. Dr. Chen's groundbreaking contributions have been widely recognized with over 60 peer-reviewed publications and numerous prestigious awards, including the Lou R. Wasserman Merit Award and Sybil B. Harrington Scholar Award from Research to Prevent Blindness. With that, please join me in extending a warm welcome to Dr. Chen, who will deliver her talk titled Fighting Blindness, Lessons from CRX Research. Welcome.
Okay, thank you, Sreha, for that nice introduction. Thank you, Chris, and for the invitation for me to have the chance to present here and then learn a lot of things from you. And so I will start this presentation with the introduction to bring everybody. It's not forward. Okay, just to bring this, to bring everybody on the same page. So here is the human retina, showing the picture of the human retina in the back of our eye. So you can see this organized in 3 major neuronal cell layers. So my lab is interested actually the studying the rods and cones of photoreceptor cells. Those cells are localized to the top here, outer nuclear layer here. And then you also see these photoreceptor cells actually produce the subcellular structure, so-called the outer segment. So imagine light comes from the bottom slide, and first will be absorbed by the photoreceptor outer segment, and where it converts to the neuronal signal, then transfer to our the inner retina, and then get processed and further transfer to the brain through the optic nerve. So there are 2 flavors for the photoreceptor cells. Rods account for 95% of all photoreceptor cells in our body. So it's responsible for night vision And the cones count for 5% of the photoreceptor cells. And in humans, there's 3 flavors, the short wavelengths, middle wavelengths, and long wavelengths cones. And the cones are responsible for color vision and our vision acuity. So these photoreceptors are essential for our vision, and they're also highly vulnerable to the environmental and the genetic insults. And there have been many diseases associated with the photoreceptor problem, so-called inherited retinal degeneration, or IRDs. So I'm showing you 3 major examples of those. And here is what normal vision look like, and I mean, for this picture. So then there's a disease so-called retinitis pigmentosa, which is a rod-centric disease. You can see the peripheral retina, the rods degenerate in the peripheral retina and leads to the tunnel vision in the end. And in contrast to this, and there's a disease so-called cone rod dystrophy. And it's a cone-centric disease. So the patient experiences color blindness and lose visual acuity. So importantly, those 2 kind of diseases are progressive. So it occur kind of any time during, in the life. And so but it's a congenital blindness disease, so-called Leber congenital amaurosis. So mainly it's due to the developmental defects that mainly affect the cones and also rods. So, so there have been more than 250 IRD genes identified, and each is linked to 1 or 2 diseases. But there's one gene actually that is linked to all 3 diseases, and mostly actually in the dominant fashion. That is a CRX. So the CRX mutations currently there's no any treatment available. So in the past 26 to 27 years, I've been really working on this issue and trying to understand CR disease, develop a treatment. So what I'm going to do today is to talk about the 3 parts. First, I'm going to talk about the biology of CRX, and then go into the understanding CR diseases, finally briefly describe the treatment strategies. So let me start with biology of CRX. By the way, if anybody want to have questions, I could just disrupt and ask a question. It's okay with me. Okay. So back to my postdoctoral work with Dr. Donald Zak at the Johns Hopkins University. And at the time, we were interested at what makes the rods as rods, cones as cones. So we know each cell type expresses a set of specific genes. And so, for example, rods express the visual pigment rhodopsin, but not cone opsins. And the cones is the reverse, express the cone opsin, but not rhodopsin. So this cell type specific gene expression is important because underexpression or overexpression certain photoreceptor genes can lead to photoreceptor degeneration. So the time we really set up thinking about how these cell type specific gene expressions regulated. And so speak about gene regulation, we know in general, and it is controlled by the regulatory DNA sequences, mostly located immediately upstream of the gene. So at the time of the Don Zak's lab, I have been using the legacy reporter transgenic mouse assays to map those regular DNA sequences. For example, if you look at the rhodopsin, so the upstream sequence contains this so-called the promoter region, just look at the 200 base pair upstream, and then that is sufficient to drive rhodopsin expression in the photoreceptor cells. However, to achieve higher level expression, it needs enhancers or located more upstream. There's one there, and that's two upstream about 2,000 base pairs upstream the rhodopsin site. So in addition, each of those large regulatory regions also contain the so-called cis regulatory elements. And so those elements is small the motifs that mainly is the transcription factor binding site. So it is the interaction between the regulatory DNA sequences and also these transcription factors that controls the photoreceptor gene expression. So at the time, say, okay, what are the transcription factors that are responsible for rhodopsin gene expression or photoreceptor gene expression? So at the time, I set up a fishing expedition. So basically, using the yeast-1 hybrid acid and with the bait, with the rhodopsin promoter cis-reactive element. And then I was trying to pull out the transcription factors from a pool of retinal transcription, retinal protein. So it turns out, and I cloned a number of homeodomain transcription factors. One of this is CRX. So what is a CRX? CR stands for Conrad homeobox, or or CRX. And it actually, the CRX gene was cloned and first reported by 3 different labs, and shown in the bottom. So so CRX is expressed by both rods and cones. And once expressed, it binds to the promoter enhancer of many photoreceptor genes. Once it's bind, and this is showing actually some consensus site for CRX. So once it's bind, it activates target gene expression, and it controls when and how much each photoreceptor gene is expressed. So as we talk about the CRX mutation associated with the blinding mostly dominant blinding retinopathies. So here is a CRX protein look like, and it has a homeodomain near the N-terminus responsible for DNA binding, and also contains activation domain, the C-terminal portion responsible for transactivation. So if you look at the 3D structure, so this is the AlphaFold model. You can see the middle here is the homeodomain. It contains 3 alpha helices to contact the DNA. So mainly through this, the helices 3 contact the major group, determining the binding specificity. So, but the C-terminal portion is really, there's no structure identified. So it's intrinsically disordered. So how CRX works? So in the wild type retina, when CRX made, it binds to the photoreceptor gene and turn on transcription in a certain time period, so that meeting the normal vision. So, but in the CRX knockout mouse, this process cannot happen. So that leads to the photoreceptor gene cannot be expressed, so leads to defective vision. So here's the picture of the histology. You can see that the 1 month, when we compare to the wild type retina, CRX knockout mouse retina shows still developed outer nuclear layer cells, but those cells are undifferentiated and they lack the outer segment, and there's no visual function, so undergo fast degeneration. And so, but the importantly to know, these phenotype has a recessive inheritance, but many of the human mutation cause dominant disease. So that's bringing the 2 part. The second part is the mechanism how CRX mutations cause disease. So I will start with some clinical features. So we're carrying out the natural history study for the CRX disease. So here is my colleague, so the Dr. Rajagopal, and also a medical student, our clinical team. And so this is our number 1 case. So there's a 14 years old white male walked in our clinic. He had no response to light at 3 months or after. So he was diagnosed as Leber congenital amaurosis. And so he has a severe nystagmus. And then this has made hard time to image his retina. So, but we were able to get some fundus photograph showing here. And first I'm showing the normal one. So you can see some structures, just follow that's the basic scientist. And we show that here is the optic nerve, blood vessels, and this region is the macular region. So here is a picture from the patient. You can clearly see, even without a doctor's eye, you can see the troubles in this macular region. So it described, the clinician described as an optic atrophy, attenuated vessels, and pigmentary changes. So then they sent to genetic test. Actually, they came out this, is a heterozygous mutation on the CRX. It's still called the Y position 191 amino acids and has a delete one nucleotide. Okay, so this is only one example. And so right now there are more than 300 human mutations being reported in the ClinVar. So I mapped some of those disease-causing mutations on the CS protein. You can see that these mutations distribute in both DNA binding and transactivation domains. And so what's interesting is the homeodomain mutation, most of the missense mutation, just change the single amino acid. And then on the C terminals, those are frame shift, maybe you have a delete one, insert some, and indel, and cause a frame shift and hit the stop codon, premature stop codon, and then basically stop there. So, basically, so how do we, why these CS mutation cause distinct diseases? To address this question, and then so we have been this kind of showing of our approach. So first of all, I talk about that we have natural history clinical study to understand the phenotype genotype correlations, and then we take these mutations and trying to understand how they cause disease, mainly the cellular molecular mechanisms. For example, we carry out the high throughput in vitro biochemical studies to understand what the impact of the CS mutations on the CS DNA binding and transactivation activity. And then we divide those mutations into different classes, make mouse models, and carrying representative mutations, and characterize the phenotype. Of course, that mostly the morphological functional molecular signatures. So basically, more recently, we started developing the human cellular mouse model, the retinal organoids model. So from this, hopefully we can learn the core mechanisms, and so we can develop mechanism-based therapy. Currently, we actually focus on the AAV gene therapy, which I'm going to talk about later. Okay, so for today's purpose, I'm going to focus on the mouse models. So maybe hopefully in the future, I can make more progress on the cellular model. Okay, so back to here, that's the mutations. So we, over the years, last 10 or 11 years, we have actually made 7 mouse models. 5 of them have been published. There are only 2 are still ongoing. So those are the circle ones, okay? And so this is actually the work started from Nick Tran, my former student, and followed by the Philip and Yiqiao Zheng. So they published a number of these papers. So I'm going to summarize 11 years of their work just to show what's the bottom line, okay, first. So first of all, they found the different mutations called disease by distinct mechanism. And as example, if you look at the homeodomain mutations, so these mutations somehow impact the CS ability to bind the DNA or change the DNA binding specificity. That causes misregulation of photoreceptor genes. And so that's homeodomain mutation. So the truncation mutation we called, and you can see that each of those mutations cause a truncated CS protein, and that they still maintain the homeodomain, and so still combine DNA, but they fail to do transactivation. That's caused misregulation of photoreceptor genes. What's interesting is that when these mutations is in the heterozygous state, so they're wild type protein there, so these mutant protein actually interfere with the wild type protein and to activate photoreceptor gene expression. So if you will, you can call this a dominant negative mutations, but that leads to the dominant retinopathies. Yes, question.
Does CRX dimerize?
I'll get to that later. Okay, and so I will first start these questions and the homeodomain mutations. Actually, your question set up what I'm going to talk about. So let's see, this is a missense mutations. They actually have 2 categories. these categories, just black and the gray, this is represent the loss of function or hypomorph mutation, if you will. So the reduced ability to the CF by DNA. But there are also the color dots represent dominant mutations. So, and if you're looking at the structure, I mean, so for today, I'm going to focus on 3 mutations that localize to the helices 3 that determine the DNA binding specificity. And those are the 3 here. So that including the loss of function mutation R90W, it is associated with the recessive liver congenital maurosis. And mainly is we show that biochemically this is a loss of function mutation. And then these 2, the dominant mutations, EADA cause dominant cone rod dystrophy, and KDN cause dominant liver congenital maurosis. So KDN is much more severe than the EADA. So at the time we had a hypothesis, each of these mutation alters DNA binding specificity and regulatory activity. So to test this hypothesis, my former graduate student Yi Qiao Zheng and carry out these assays. You can call this, if you will, is a multi-omics approach. So basically what she does is was the so-called spec-seq or COOP-seq. Those are terms, but think about it, this is a sequence-based gel mobility shift assays. Mainly, so for example, I give you an example of the spec-seq. You can see, and we made the double-strand oligo libraries. You can have this CS binding site, the 6 nucleotide. The TAA is fixed, but you can see the position 4 to 6 is random. And so that's the library. So then each will purify the CS homeodomain, either it's the wild type or each of those 3 mutants. Then after incubation with oligo library, she ran the gel. And then EMS gel, she can isolate the fraction of a bound or unbound fraction and then send to Illumina sequencing. So, okay, so they got that showing the binding changes, but what about activity? So she actually carried out the reporter assays mainly. So there are 2 types. One is a low throughput. You can do the luciferase reporter assays. Traditionally, that's in our lab in HEK290 cells. And also she carried out the massive parallel reporter assays in the retinal explant. So you can call this ex vivo. And most importantly, she could correlate what these findings, biochemistry findings, to the consequences in the non-PN mouse models to see how those changes affect the in vivo cells binding, chromatin accessibility, gene expression, and also the consequence on retinal morphology, function, and behavior. So I'm going to start with non-PN mouse models. Okay. So let's start here. So we made the non-PN mouse models. So again, we here with histology, the first one's wild type, and the second is heterozygous mutants, and then homozygous mutants. Okay. So if you look at the R90W, like I said, it's a loss of function mutation associated with the recessive LCA. You can see that the homozygous mutant contain the outer nuclear layer, but they do not, they do not elaborate outer segment. It's blind and then undergo fast degeneration, just like a CS knockout mouse. But heterozygous is fairly normal. You can see good outer segment here. And so, but in contrast, the 2 dominant mutation here, you can see, of course, the homozygous mutant shows very disrupted nuclear structure shown here, whorls and rosettes, but has also shows abnormalities. Of course, the KDN is more severe than the EADA. So we can see the wavering here. So basically from this, and we thought we have a mouse actually resemble the phenotype reported for the human patients. So, and the next question will be, what is the mechanism? How these homeodomain mutations, causes by altering DNA binding specificity? So to do that, the Yi Qiao carried out a spec-seq. So then he basically generated called the energy wave matrix. Okay, so basically to show the consensus binding site. And I'll orient to you this pattern here and this panel. So on the x-axis, I show different positions. And remember, our library is, changing the ends, only the random on this position 4 to 6. And then on the y-axis, I show negative energy. So in such way, you can see that anything above the 0 will be favorite, preferred binding, and then residues, and then anything below the 0 will be unfavored. So based on this, she generated the position. I mean, it's not position, energy weight matrix. You can see is TAATCC. Okay, so that's consistent with the previous reported position weight matrix or ChIP-seq assays things. So what about mutants? So here's R90W. And you can see actually R90W, if you have a higher concentration R90W, it can bind this. So they still prefer the wild type preferred sequence here. But it has to spend a relative higher energy to bind. So in a sense, it's a reduced relative affinity. And so that's a loss of function mutation. What about here, EATA? So if you look at ETA, it still prefers a similar consensus site, but it spends less energy. So meaning they, even in the low concentration, they can bind to this kind of sequence. So it's increased the relative affinity. And okay, this EATA. What about KDN? And the KDN completely changed the binding specificity. Instead of the GC rich in the position 5, 6, now it's 80 rich showing here. So this is mainly altered the motif preference. Okay, and so how do these biochemical changes impact the in vivo binding and gene regulation? So basically, Yiqiao performed the ChIP-seq and the RNA-seq in the bulk, in the mouse models. So, and I'm just going to summarize what she found. Okay, so basically, when she looked, this is a wild type data. So we see the ChIP peaks here, and then under that is the consensus side from the ChIP peak that's enriched to this TAATCC motif, similar to what we determined the SPEC-seq. So that is required for activated photoresist gene expression, again, using RAW as a model. So you can see the activate this expression something between the P5 and P21 and during the postnatal development in mouse. Okay, so, but what about this loss of function mutation? You can see we cannot detect any binding, in vivo binding. So even though in vitro, they still combine the sequence, but there's no ChIP peak showing here, mainly cannot bind to chromatin template. So, and as a result, the fail to activate photoreceptor gene expression, and so it's essentially a loss of function mutation, and that's what associated liver congenital cirrhosis. And then next one's interesting. So the KTN. So it doesn't bind to the consensus site, CS site motifs. It's gained ectopic ChIP-seq peaks with this consensus site, the TAATA. Okay. So obviously, I mean, this is a different. This is actually resemble the progenitor cell transcription factors like VSX2 and LHX2 and RAX, if you heard those. And so as a result, this is antimorphic, so they cannot start differentiation. That is associated with LCA, either dominant or recessive fashion. And this one's interesting. So the EATA, on the other hand, it still combines to the wild type site, even they show these peaks and then with this consensus site, but it appears to be hyperactive. The reason I'm saying that is they seem to bind early and then activate the photoreceptor genes in early stage. Instead of the P5, now it start rhodopsin expression, the P3, and it's amazing. And then so this is but it just cannot, they show the early shifted this curve to the left, but they cannot hold it in the later stage, so they dropped. So this is associated with dominant congruent dystrophy or recessive LCA. So from this study, And she concluded that the precise target binding and regulatory activity are essential for route cone development. So the homeodomain mutations can impact target binding in different ways. Of course, this will provide some the insight into the treatment strategies. I'm going to elaborate that later. Okay, so but there's still we were puzzling. There's one issue on this model. These are the model. So for EADA particularly, so why it's dropped later development? So it's a, it's hyperactive. Is it really a hyperactive mutation? And I'm telling you, it's partly right, but it's not the full story. So what happened is, so Yiqiao looking at the photoreceptor gene, what does she call this CF-dependent activate gene? There are actually two categories. So one category contains this monomeric CF binding site only. So that's only count 1/3 of those genes, total photoreceptor genes. But if you look at the other 2/3 of those photoreceptor genes contain not only the monomeric, but dimeric sites. Okay, so assuming they can form dimers there. So what's more important, if you're looking at the changes of these dynamics, this gene expression during development, you can see the different pattern. So you can see the monomeric only, you can see the gradual increase after P7 and then plateau. But in general, if you compare the monomeric plus dimeric side, you can see the monomeric only one is lower expression in the end. And then the mono plus dimer side, you can see there's later there was an explanation or increase. So give you an example, rhodopsin. So you can see again, there's a slow, increase here between the P3, P10, but after that, there is a great increase of expression and reach very high level. And the rhodopsin messenger RNA is count about 1% of total RNA. So how is this happen? So what is the contribution by this dimeric binding site? So that's the question. So she's actually realized that CRX homeodomain is paired like homeodomain. Maybe they can form homodimer. And we call that the P3 site. What I mean P3 site is this. This half site, one half site two actually form the anti-parallel orientation. And there is a 3 space in here, okay? And so that's usually they form homodimer on this kind of a site. So then Yiqiao said, well, let's look at what are those CRs, this 61%, whatever. What are those dimeric binding sites in the retina? So look at this. So she found that, plot this as a relative binding affinity. That's based on the SPEC-C. So you can see that between those two sides, at least one of those sides, either side 1, side 2, is low affinity, okay? Or sometimes they're both are low affinity. So what that means is that in order for CR to form homodimer there, it requires cooperative dimerization. What I mean for that is binding to the first half side greatly enhance the binding of the second molecule to the other half side, okay? So then the question is it really the CRs can do this cooperative dimerization? How do mutations impact this cooperative dimerization? So to do that, Yiqiao carry out this called the CoopSeq or measure the cooperativity. Maybe you have the library contain the dimeric binding site, so the TAAT and spacer, and then TAT. So they made the two kind. The P3 library has the 3 base pair spacer, and then but the P5 library you have a 5 spacer. And normally this 5 spacer will disrupt the cooperativity. So you can binding as a 2 dimer, 2 molecules, not a cooperative dimer. It could be. So she basically incubate this CoopSeq library with a CS homeodomain, either wild-type mutants, and isolate monomeric, dimeric fraction, and unbound fraction, and Illumina sequencing. And then she calculate cooperativity index, okay? So basically, She started with the wild type. So the blue is the more cooperativity and the red, or, is less cooperative. So you can see the wild type, the half of those sides actually showing the cooperative activity. And R90W shows a very similar pattern. So not change. And just similar as wild type. But if you're looking at E80A, you can see it's greatly reduced dimerization, on this supposed to be cooperative sites. And what's amazing is this one. So you can see KDN actually gain the cooperativity. It doesn't matter what sequence is, this NNN can be anything. So they still bind, it's cooperative. So we scratched the head and say, how is those biochemical property changes leads to the in vivo thing? So what we're gonna do is to focus on the E80A, not mutants. Okay, so here it is. Before I talk about that, and then first I'm going to introduce another concept or the mechanism of CS action is to basically mediate the chromatin remodeling during photoreceptor differentiation. Mainly in vivo, the photoreceptor gene is not naked, so actually associated with the nucleosome. So the histone octamer to bind there. So I mean, basically in the non-photoreceptor cells or even progenitor cells, and these has an array of the nucleosome, and that is impressive. And so photoreceptor genes are not expressed there, but during the development, and these nucleosomes start rearranging, and then the chromatin gradually open. You could start early as a partial open, then later you get more open, such that later will be free of the nucleosome here and mediate the full level of expression. So when my former grad student, Philip, come in saying, okay, what's the role of CS in this? Basically, what he did is he did the ATAC-seq, which measured the chromatin accessibility, and the CS chip-seq. And what he found was the CS is required for this process, because in the absence of CS, this process won't occur. So the photoreceptor chromatin may remain in the closed configuration. So what about E80A retinas? So we did that, and so basically she did it, and then here we were comparing the changes of the ATAC-seq signal. So there are probably, it's number here. So more than 1,681 sites actually showing the increase the ATAC-seq signal in the E80A retina. And you can see the homozygous is a pretty amazing increase, while the hex is in between. So, and then you can see that's corresponding to the increased E80A binding. Oh, by the way, this is P14, okay? And then if you're looking at the site there, it's enriched with the monomeric site. And then if you look at the development, you can see that E80A increase peaks during the development shows the early increase and then plateaued after P7, P10-ish. Okay, so, but there are also other side, right about 500-ish, it actually shows decrease the ATAC-seq signal and corresponding to lose this E80 binding. If you look at the motif enriched here, you can see it's very what called the low affinity the dimeric sites. And those sites actually during development, you can see the pattern is slow increase, and then after P10, you see a big increase and then remains highly open. So how do we say it's really directly those dimeric sites involved the regulation? So Yiqiao carry out the massive parallel reporter assays. Mainly, she took those peaks and bound by the basically the CS wild type and the mutant, and then she made a library that contained those peaks. And this library is using the low minimal promoter as driven the DS red as reporter. So she electrocuted into the P0, the wild type retina, and also E80A retina. and then collected at the P8 for DNA RNA sequencing. Here's the result. So first we're showing you the monomeric site. You can see in general, and then these monomeric site and showing the increase the activity in the EATA, particularly the homozygous retina. And then, but in comparison, if you compare the dimeric CREs, you can see actually the EATA, it's reduced the activity on those sites. Okay, so that seemed to provide a direct evidence that there's a meaningful that's the regulatory activity, at least in the non-chromosome template, but correlated with the in vivo endogenous gene result. So here is a model. So in the wild type retina, you can see the many of the photoreceptor genes contain both monomeric, dimeric binding site. So during the early differentiation, the monomeric one was bound by the CRS molecules, and then slightly turn on gene expression, and later, and then the CRS concentration really high, so they can bind to this dimeric binding site, and then to drive the photoreceptor gene to a very high level. And in EATA retinas, on the other hand, so the EATA combines to the monomeric site early and or more activity. So this is a gain function. But later, these mutant protein cannot bind as a homodimer to this dimeric site. So it's mainly the later is a loss of function. Okay, so both gain function, loss function produce this phenotype. So yeah, so this is what happened to the early shift left and then later dropped. Okay, so future directions. So those are based on the raw data, maybe the whole bulk RNA-seq, the seq data. So what is the how do we understand why there's cone rod dystrophy? So basically, if we look at the cone genes, majority of the cone genes, 80% of them actually contain the monomeric plus dimeric site. But in compared to the raw genes, only 60% contain the dimeric and the monomeric sites. So that's why we think of the cone genes is really reduced, start misregulated. So that's cone rod dystrophy, but this is just our model right now. So we have to look at later. So we also need to test the therapeutic strategies. And to correct this kind of a mutation effect, we need to have some kind of a combined gene augmentation plus knockdown, or somebody says knockout, replace, that kind of a strategy. Okay. So anyway, so I'm going to switch to the frame-shift truncation mutations. Okay, so should I move on? Yeah. Okay. Here's the frame-shift truncation mutations, and many of distribute here. So I have some examples here showing this red. And so they are associated with a wide variety of a range of a phenotype. So at the time, and we thought, okay, so that's the model, and the mutant protein lack the transactivation domain, still combine DNA, and meanwhile interfere with wild type function. And so we tested to test this hypothesis. Nick Tran, a former my student, actually made this mouse model, and that's 11 years ago. I can't believe it. So basically, it's developed this the phenotype like a dominant cone rod dystrophy, just similar to what reported in the human. So patient. So basically, you can see that at 1 month. If you staying for the cone arresting, you can see we lost the cones and compared to wild type. You also see the raw degeneration occur at 3 months. You can see here start at 3 months and it start degenerating. And that's fits the ERG data too. So, and this is interesting, but what's the mechanism? We also, to our surprise, when we perform the Western blot, so just to orient you, the full length is showing here, and the truncated protein is here. So if you look at this Western blot, you can see the product is made by the mutant allele, not only made, but made twice as much. So this is a quantification on the right. So, and made twice as much as the wild type. So I call it unfair competition. Basically, you made a lot of mutant protein to compete with wild type. And so is this overproduction of the mutant allele? I mean, it is a part of a dominant negative mechanism. So to do that, we actually tune down the mutant expression, mutant allele expression, to normalize to 1-to-1 ratio, like here. So, and in that sense, this mutant mouse actually shows 1-to-1 ratio. Now the phenotype is much less, severe. So we essentially seem to cure the disease. So that makes a really encouraging thing. What about if the potential therapy, we just trying to find a way to greatly increase wild type to mutant ratio that would help, right? Benefit. Well, but at that time, I thought, okay, that's only based on the one mutant. What about other mutations here? And so we're still characterizing this, particularly in this mutant. So I'm not going to show the data today, just to say that this mutation actually produced a very similar phenotype as E1CTD2. If anything, it's actually even more severe. And then, but importantly, they also show this kind of things, overproducing the protein and RNA, actually. And so anyway, so that, bring me to the thing. The third part is, can we develop treatment for this kind of disease? So basically, and I thought at the time, when we want to develop a gene therapy. So, but mostly, as you all know, the gene therapy treating the recessive disease, and so our challenge is the CS mutations and dominant residence, inheritance. We really have to really boost up the wild type of mutant protein ratio. But in the meantime, CS is a regular protein, so this is dose sensitive. So what we do, so basically, I came out this as a tunable CS augmentation. What we want to do is I developed the TED-off system. So basically, we can make this is a cells contain the mutant heterozygous mutation. So if we can introduce the TED-off AAV gene vectors and into the cells, we can greatly increase the expression of the wild type CRX. And in the meanwhile, if we overproduce, we can use the doxycycline system to tune down to reduce the toxicity. And that's at the time, that's the idea. So will this work? So I decided, okay, let's try it. It's just a proof of principle. So what we did is we packed those TED-off system into the AAV5 vector. So and again, we are using the D2 mouse model. And so we injected this AAV virus into the subretinal space at 2 weeks in the mouse, and then we harvest at 6 weeks. And the reason that too short is that we know that 6 weeks, this D2 mouse will have no cones or very little cones left. So we did a whole mount stain for the cone arresting as a marker. So you can see, and I marked the transduced area with red and non-transduced area in the black. You can see, and they're only without transfection, transducing, and you can see there's very few cones left there. But with the transduced area, you can see with the AAV treatment, you can see a lot of more cones there recovered. So, and we were very happy about that. And then we said, okay, let's do 3 months. But the 3 months we see these cones, recovered cones, still undergo degeneration. And later still, still progressive degeneration, we just rescue a little bit more. So we need a better efficacy, better result. So also at the time I thought, well, we see as a rare disease, we need to develop something, one strategy fit our mutation. So we came up with this. What about combinational? So we do the gene augmentation plus knockdown. So basically the same cells, here has a point mutation there. So if we introduce CRX in the meantime, and this is augmentation, then we also remove this endogenous, the mutant and wild type allele. So that hopefully that approach will be more effective. Does that work? Now it's a proof principle experiment again. So we're looking this vector. So we have a photoreceptor promoter driven flag tag CRX. Then we have U6 promoter driven the SHR against the endogenous mouse CRX. Okay, so we pack this in single vector. So that would be injected into an extreme case that we call the D2D2, which is most severe homozygous mutant D2. And we know that at 1 month, the rod degenerate also, and then no cones. So we did a P14 injection and harvested 1 month and to look at this, okay? So what we found is that this is the untransfected area. As expected, they start degeneration. So very thin outer nuclear layer here. This is the same retina in the AAV infected area. You can see greatly increase the outer nuclear thickness. And then we recover the cone markers here again. So this is a really only preliminary. So we're repeating experiment and looking at the later ages. And so basically that's preliminary, but I think it's very promising and interesting. Okay, so with the time I'm going to just summarize. And so basically I show you, and for 26 or 27 years of CS research, we took this approach from clinic to laboratory. And so we have now gained more understanding of how CS mutation causes disease. And now we are to see whether we can, this mechanism we learned will be applicable to the human patients. So basically we use a human cellular mouse model from the IPS, and we're still developing. Hopefully I'll get result later. So we're hoping that the treatment strategy developed could, in the end, we end to some clinical trial to restore patient vision. So with that, I'm going to now acknowledge that people did this work. So this knocking mouse model studies actually started by Nick Chuan and former, Philip Ruzuki, I mentioned, and then followed by the Yiqiao Zheng. And then she now is a postdoc fellow in MIT. And both Nick Chuan and Philip are now faculty, tenure track assistant professors. And they have their own R1s and work on different fields. And then our study is still now followed by the Qi Sen and the Xiaodong and the team, So I'm going to stop. So just to acknowledge our funding agency and also not only the NIH and thanks to the research prevent blindness and also our private donors. And then this is our resources at WashU and help me to do the translational research. With that, I'm going to stop here and to take questions. Thank you for all your attention.
Hi, good morning. Thank you so much. That was a really interesting talk. This is slightly tangential, but I was curious in some of the earlier slides you showed the histology of different mutations in the homeodomain, and you showed that in some of them you started to develop rosettes, and in some of them you didn't.
Yeah.
So when I think about rosettes in the clinical context, I think of the RPE disappearing and then the outer segments of photoreceptors trying to find a place that's a more hospitable environment. But what you've shown is that the outer segments don't form and the outer nuclear layer is there. So do you have any ideas as to why the rosettes or how they might be related to CRX?
That's a good question. We also scratched the head. What is the world's rosettes thing? So we looked at the expression, like we know in mouse, that's probably the phenotype particularly to the mouse models. And then we know the mutations in NRL, so or NR2E3 generated this world's rosettes. But if we look at the NRL, NR2E3 expression, and particularly for EATA, they're there. So it's not losing NRL or NR2E3 expression. So we do not know how is this happens to the world's rosettes. This is a lot of questions that people still trying to understand what's the mechanism.
Thank you. If I could just ask one more question too. If we use a CRX GFP stem cell line for producing retinal organoids, and you've shown us that the N terminus, which binds the DNA, is fairly rigid, but then the C terminus is fairly flexible and unpredictable. Do you think that if you put GFP on top of that, that might alter its ability to regulate expression of photoreceptor genes?
Oh, so you fuse the GFP to the CRX?
Yes.
So I would say C terminus is very important. And then if you fuse the GFP there could be some changes of activity. And but I cannot, basically there's an intrinsically disordered structure, and there are requires some kind of amino acid composition, for example, charged residue versus the hydrophobic, protein or romantic residue, something like that. So there is a mutation scan result published. Okay, so but I don't know how is that going to affect the fusion protein. I believe there's some activity changes. If anything, you mess up the C terminal CRX.
Yeah, Shiming here. Yeah, it's a very nice talk, and thanks for such detailed study. So I have two small questions. One is I noticed some of the mutation in the binding domain, homeo domain, and then you said some of the mutation have both recessive and dominant phenotype. I'm just wondering what's your thought on the same mutation have two different inheritance there?
So I think that for the loss of function mutations, mostly for loss of function mutations, actually mostly the particular R90W is only associated with recessive disease. But they're hypomorph, and so R40 or R41W, those kind of mutations seem to have some dominant phenotype, but this is less severe, maybe late onset dominancy, late onset disease or mild disease, if you will. So I still don't quite understand why, how those mutations cause disease.
So when you refer dominant and recessive for the same mutation, those coming from human genetic observation or from the
From
mouse?
mouse, okay.
From the mouse.
But for that, I think raised a very good question, because I do have a family that has carried the same mutation, a dominant mutation, but one of those are making mouse model, and they have family members. They actually show some kind of a big phenotype variability in terms of age of onset, even the phenotype variability. So some males start at peripheral, then go to central, and the female goes from central then to peripheral. So there are genetic, I think there's a heterogeneity could be contributed by the environment and the genetics or genetic makeup. So we're still trying to understand that. So also depends on the expression of a mutant versus wild type allele. So that we're trying to use the retinal organelle model to see what happened, to those heterogeneity.
So my second question is regarding, it seems from the data, the CRX dosage seems quite important, right? The amount of CRX that we're expanding. So when you have the therapy strategy where you all express augment therapy with CRX, you notice that, later on, this initial rescue and later on just, sort of progressive degeneration. Even in your last slide where you use SHRA to knock down the endogenous.
Yeah.
And I do notice, like, for example, the cone position is not correct, right?
Yeah,
it's just like everywhere.
perfect.
So is that, right? Yeah. What's your thoughts on that? I mean,
Yeah,
it's just only because dose is sensitive, but the augmentation will continue to be an issue.
Yeah, I think that's a good question. And then many of the gene therapy also had these issues. Early, they seem to be very encouraging. Later, long-term effect, efficacy still reduced. So we're currently developing something, not only gene therapy, but you also some kind of neuroprotection drugs, because imagine the cells are sick for a while. So then I mean, in fact, many of those mutations, the clones, you don't detect a marker, but the cells are there. Actually, we do the lineage tracing, they're still there. It just not function, so they're sick. So even you put a gene therapy vector there, they may not fully recover, so similar, I think we can discuss other approach. So you correct mutation, but if the cell sick, so we need some kind of combination of gene therapy, genetic correction and neuroprotection or energy boost, whatever it is, to prolong the therapeutic effects. So.
Okay, we have a question from the Zoom. Yunluk Zhou, are you ready to ask?
Yes. Hey, Shiming, fantastic work and talk. I definitely learned a lot. And I have a very quick question. I've been studying retinitis pigmentosa since I did my postdoc with Connie. And because CRX mutation, one of them is linked with dominant form of retinitis pigmentosa. And from our understanding, it's usually the rods got affected first. So have you or anyone else made a knocking mice with that particular retinitis pigmentosa associated mutation and found any phenotype?
Okay, so that's an interesting question because I didn't show, do I have? So I have one mutation and we know the patient, okay, I don't know where is the truncation mutations properly. Basically, so I have example here. So if different dose, so initially we have a patient coming to our clinic saying, okay, diagnosis as RP. And so we sent it to genetic testing, and then it came back to the CS mutation, one of those here. And then we said, okay, and then this is RP. But based on my former mouse model, I predict it should be cone rod dystrophy. So I said, wait, this is not RP, it should be cone rod dystrophy. And then the clinician said, okay, I'll close watch. 2 years later, it developed both eye, in the macular region. And indeed, initially they have peripheral lesion, but then they switched to central retina. And then the diagnosis now changed to the cone rod dystrophy. So now this patient, 5 years later, has no vision. So basically legally blind.
All right, so I will ask my question.
Can I do a follow-up?
Slide that you were showing originally, and R90W mutation. It appears that with the loss of the outer segment of the photoreceptor, outer, maybe inner segment as well, you have some changes in the inner retina. Is it my correct assessment?
Yes, if you have early, once you have degeneration, right? So you will affect the, inner retina, the whole wires, so. Yeah, so early, just like a CS knockout mouse, they have an early start with ONL degeneration, and later there's a remodeling in the inner retina. So that really makes the therapy so very challenging. You really need to start early, not late. Yeah. Yes.
Hi, Dominic here. I wanted to ask you about these homeodomain mutations. Do those mutations cause like any strong, like structural changes to the protein? Did you try maybe to do any simulations or any alpha fold foldings of this domain with the DNA? How do these mutations affect the actual domain structure?
Yeah, I mean, that's a good question. We haven't done that, but my colleague did the mutation scan. So basically changed every single amino acid to other 19. So she found those anything loss of function, mostly loss of function, is located at the surface contact DNA. but there's no simulation yet to show they change the alpha helix structure for the single amino acid, right? Changes.
Hi, a wonderful talk. I wanted to ask you about the lysine 88 to asparagine mutation. You showed that it's actually more cooperative than wild type, but it's also antimorphic. So I'm wondering if it's kind of dragging the wild type towards off-target regions, and you're seeing more ectopic expression for this particular mutation. And in that sense, would gene augmentation be helpful, because it's again, it seems to be cooperative, more cooperative, and would have a negative dominant impact on wild type. I also wanted to ask about CRX in terms of treatment. The treatment window, because it's so important in early development. What would the window of administration look like for this sort of master regulator?
Those are all good questions, and I really did not talk about the KDN. So here's the problem: the KDN, I say the antimorphic because they're it doesn't bind to the CR's photoreceptor gene. Any genes, regular regions, but it goes to the wrong place, right? So this actually, during development, if you I gave a little bit example. So when you do the development, you want to do differentiation, you need actually two things. One, you activate the differentiation program. Two, you have to suppress the other program. For example, suppress the progenitor program. So what if you somehow the KDN is a progenitor cell binding site? So they don't shut off the progenitor program, and then your differentiation cannot go. So there's two sides. I didn't mention that. So in that sense, this is very toxic. So basically, that's why LCA so antimorphic. But if you want to correct that, if you want to do the therapy, you mainly have to shut down. You have to knock out this mutant alleles. And then, so gene augmentation won't help, because the heterozygous mouse, they show the similar things. And yes.
Hi, Dr. Chen. I'm Xuan from Dr. Rachen's lab. So thank you for your wonderful talk. I have a question about the frame shift mutation part. So it seems like the truncated protein competes with the wild type protein for the binding site. So I'm wondering that if what's the fate, if we can determine the fate for the truncated protein, such as targeted for proteasome degradation, so that the wild type allele can produce a sufficient protein for the functions, and we don't worry about overdose issue for the augmentation therapy. Thank you.
That's an interesting idea, so we have to do the specific. So we know that the truncation mutations, it's very toxic. And then as time goes, I don't understand that mechanism. So the early on, you have a small ratio of mutant to wild type. As time goes, all the remaining cells become mainly produce the mutant allele products. So that's very bad. So I don't know, I'm open for any suggestions on how to target the mutant allele specifically, truncated allele, not the wild type allele, because this is in the last exon, so they don't undergo nonsense mediated decay, but I'm pretty sure we can try to find that. So my future study direction is to understand why this RNA proteins overproduce. Is something related to the RNA binding proteins, or we could try to understand that mechanism. So it's an ongoing. Thank you. Yes.
Hi, it was very, very interesting and exciting talk. Thank you. I was wondering if you have looked at any properties that allow those homodomains, those proteins dimerize, interact, and whether those mutations affect this property. And also, one of the thoughts is, have you thought about activation domain impact on interaction between the proteins since they are anti-parallel? Well, anyway, I can see the potential here.
Yeah, well, we only done this, we think there's a homodimer. Well, I mean, are you talking about the heterodimer with some other?
Homodimer.
Homodimer, okay, so.
But how do they do that?
Yeah, we needed to, okay, I don't have the procedure to target that one. So I think we need to measure on-off rate or something like that. So this is our relative affinity changes. So, but that's interesting. And we also for truncation mutations, we do not know whether they really affected DNA binding activity is the same as the wild type. We do not know yet. So, and that's difficult, especially in heterozygous. So how do you to do that? We need a tag the mutant versus the wild type and to see the chip whether they combining there or not, or how it works. So it's challenging. So.
Hi, Dr. Chen. Thank you so much for such an interesting talk. So there has been work that suggests that CRX interacts with different sex chromosomes, and that would account for the discrepancies that are seen in different patient populations, and specifically in phenotypic demonstrations in both women and men. So were you able to observe any of these, or were you able to account for when you were studying these different CRX disease models, or even in treatment between these two different genders?
Let's put this way, if we do the mouse model, the male or female, we don't see the difference. So I think there's probably the genetic makeup, or something that human specific ones. But because CRS is a rare disease, CRS, all diseases are rare. So we don't have enough patients to target the sex difference. But it's interesting thoughts. So.
Hello, my name is Rafał. I'm postdoc in Chris's lab. Exciting talk. I like this so much. I have a question. Are the other aspects of physiology affected by CRX besides the eye, for example?
CRS is particularly expressed mostly in the photoreceptor in the retina. And I know it could be expressed in the pineal gland and maybe involved in the circadian entrainment or something like that. But we don't have a core evidence to show that patient carry the mutation has a disrupted circadian rhythm or entrainment or any of those. So again, it's rare disease. We thought we could look at it, but we haven't. So, and our mouse model are built on the C57 black. So they don't have a pineal gland. So there's it's not the proper model to study that. Yeah.
Thank you.
Hello, Dr. Chen. Thank you very much for your interesting talk. It seems that accessibility of this binding site also has some role in all this. Are there any thoughts about therapy that would affect this process, like chromatin remodeling or like inhibitors of histone acetylase or something?
Yeah, that's a good question because CRS has two mechanisms of action. One is chromatin remodeling. Two is true transcription activation or regulation. So we try to say, can we just using how the mouse model put something chromatin remodeling drugs, but those are not specific. They can say, can we do the TSA increase the ability of chromatin opening? But if you have CRS mutation, that true transactivation will still disrupted. Just like we did in the massive pyrororeporter acid, this is the outside chromatin, the episomal plasmid. So we still show the defect. So I think it's difficult to see alone if you have chromatin remodeling drugs, but we still need some genetic correction. Yeah.
We had a wonderful conversation and great presentation and questions. And let's finish with the best one from Vladimir.
Thank you, Chris. Beautiful work, Shiming, and I particularly love, and I think it's very impressive to see how you have stuck with CRX from the initial identification of the gene to then figuring out the disease-causing mutations, understanding the mechanism of the disease, and then finally coming full circle and developing therapies. I have two questions. The first is, you showed us that about a third of the motifs are monomeric and then the other 2/3 are dimeric. Is there any systemic difference between the genes that are photoreceptor genes that are monomeric versus dimeric CRX motifs? Is there something special for one versus the other group?
Well, I think that's a good question, because if you look at most of these, the genes contain a dimeric site for the ROR gene. You can see those are the highly expressed ROR gene, like rhodopsin GNOT, and then a couple others like ERB or something like that. So the higher expression genes are almost sure to contain this, both dimeric.
So that's just a mechanism to boost expression, presumably.
Yes,
okay.
yes.
Okay.
Yes.
And my second question, somebody brought this up already, but the therapeutic window. So I noticed that you treat your mice at P14, which is still for mice, it's still during the development of photoreceptors, but for humans, I guess development happens in the embryo. So how do you see ultimately the therapy being delivered to patients that have these mutations?
Yeah, so this is gene therapy general phase the question. So if you have the cells, you can do the gene therapy. If you don't, you have to do others in the procedure. So our gene therapy would target this patient has a cone rod dystrophy at a later onset. I mean, for example, our number one patient, the retina is already pretty bad. I don't think it's good candidates for the gene therapy, and so that's pretty sick, And but that raises another question. In mouse, actually, when we do the truncation mutation, we did not only the P14, we did the P21 or later. So basically, it's early therapeutic effects much more, effective than the later ones. But we still can recover, because I use a transgenic mouse to put it into the inducible gene to show that we can do that later, like 1 month even, for D2 mouse. But again, the therapeutic effects goes significantly down when you have later. And then as for patients, so like I mentioned, the patient where we're going to target those a little bit later, adult onset cone rod dystrophy type, So many of those actually start the disease phenotype at their 20s, 30s, something even, we can go to teenagers, but so really early one, I think is too toxic to, too sick to target.
All right. Thank you so much for your presentation. Thank you.